Hello friends, welcome back to my YouTube channel Astin Padasale. Currently we are discussing the answers for this Barc OCS Biosciences examination. This is a part 2 video. So first question is gramicidin A inhibit the ATP synthesis by. Okay, so they are asking the mechanism by which this particular antibiotic will be inhibiting the oxidative phosphorylation. Okay, so the correct answer for this particular question is that it will be disturb the osmotic gradient. Okay, so this we had revised lots of ETC inhibitor. At the same time, you should also know about this uncouplers of this oxidative phosphorylation and ETC. Okay, so here if you check the mechanism about this particular gramicidin A, it is coming another category called channel forming ionophore. Okay, so it will be making the biological membrane to be more permeable to specific cation. Okay, so this particular mechanism will be disrupting the cellular ionic homeostasis. So ultimately the osmotic gradient will be disturbed. Ultimately what will be happen? ATP synthesis will be stopped. Okay, whereas this particular compounds are inhibited by the ETC inhibitors. Okay, like antimycin A, rotentin, like that. Okay, next question is that a CAMP is acting as a signaling molecule. So they are asked how the CAMP is acting as a cellular signaling molecule. The correct answer is that it will be activating this protein kinase A. Actually, there are three main targets will be available for this particular secondary messenger called cyclic AMP. First is your protein kinase A, next include the exchange protein, and next include the cyclic nucleic graded ionic channel. Okay, so if you take about this protein kinase A, so first message will be received by this G protein coupled receptor. Then the adenyl cyclase will be converting ATP to form the CAMP. Then it will be acting as a secondary messenger. It will be converting the inactive form of this protein kinase A to be active form of protein kinase A. Then this protein kinase A will be traveled inside the nucleus and it will be transcribing few important genes. So that's how this particular CAMP will be acting as a secondary messenger. Okay, so I think you will be getting confused with this IP3 which will be increasing the Ca2 molecule. So correct answer is that it will be acting as a secondary molecule by activating this protein kinase A. Next is Vir A and Vir G question has been asked. The correct answer is that both Vir A and Vir G are continuously, that is constitutively expressed. At the same time, both are transduced by a two-component system. That is, if you check over here, agrobacterium two-component regulatory system, they will be sensing and that will be reacting to the presence of susceptible cell. So this particular, there are two component system I just mentioned which include Vir A, which is a membrane sensor protein. Second component is that Virg, which is a cytoplasmic response regulator protein. So both together they form a two-component regulatory system, and what they will be they will be activating the other Virgin. Okay, so they are continuously expressed and they are transduced by two-component system. Next is mustard oil contamination with this particular argemone oil. Okay, they are asked the particular compound which is responsible. Okay, so the correct answer for this particular question is sanguinarine is the correct answer, and it is a toxic polycyclic cell which is present in your argemone oil, and it is an adulterant which you can able to notice in mustard oil. But the Indian government has recently announced an important test, chemical test to determine whether the mustard oil is adulterated or not adulterated. See, if the mustard oil is not adulterated with this particular argemone oil, means what it will be means, it will, when it reacts with nitric acid, it will be remain, there will be no color change. Okay, whereas in case the mustard oil is adulterated with this particular argemone oil means, then on reaction with nitric acid, it will be producing a orange, yellow to red color. Okay, so the adulterant present in the argemone oil is this particular sanguinarine. Okay, because one of the option has been given, I don't remember the rest of three option. So I have given a correct option. Next is a gene silencing question has been asked. The correct answer for this particular gene silencing related question is that organate protein is the correct answer. They are a key player in the RNA silencing, and this particular protein family, they are key player in the gene silencing pathway, and it will be guided by the small RNA. So small RNA which include a short interfering RNA and microRNA and the PV interacting RNA, and all the three RNA will be helping this particular protein to target the particular messenger RNA for silencing or destruction process. Okay, the protein which is involved in gene silencing is this organate protein. Next is cross-pollination does not occur. So cross-pollination does not occur in this particular Callistogamous flower because why it doesn't occur in that particular flower means this particular flower will not open at all. So their anther and stigma will be inaccessible. So that cell cross-pollination is not at all possible in this particular flower alone. But anyhow they will be producing seeds and other process because they will be having the self-pollination mechanism. Okay, so cross-pollination doesn't occur in this particular Callistogamous flower because they are considered as a closed flower. Okay, they don't open. That's why the anthers and sigma will be remain inaccessible at the time of cross pollination. Next is high stringency condition during a time of nucleic acid hybridization. The correct answer is there should be a low amount of cation. That is, there should be a low amount of salt concentration and there should be a high temperature should be maintained. Only the high stringency condition will be maintained so that your probes will be correctly matched to the target sequence. If it is low stringency condition means it will be producing few abnormal result. Okay. Next is there is a question regarding to nitrogenase enzyme and they have given 4 statements and they have asked to find out the incorrect statement. So I just find this particular option alone is there in my mind, that is more than 20 ATPs are required to fix one nitrogen. But we all know 16 ATPs are required to reduce or to fix one nitrogen under this biological nitrogen fixation. So this particular option is the incorrect one. So if you are given this particular option, you will be getting 3 marks. Next is with regarding to this nuclear pore complex. So first option is completely wrong. Here they had given nuclear pore complexes and transport across this nuclear pore complex is unidirectional, but it is bidirectional. So here in this article you can able to find the nuclear pore complex transport is highly selective and it is a bidirectional transporter. Second thing, it will be activating, it will be taking out this active transport, but here you can able to find there are two main mechanisms of transport to this nuclear pore complex. One is passive diffusion, another one is facilitated diffusion. Okay, so you can eliminate this particular option. Third option is that they have given large molecules can be carried with the help of a few cargo proteins like that they have given. Absolutely correct. Large molecules can also be passed, can go in or out of this pore complex because they will be having few cargo protein. Next, last option is also correct, but it is not that much correct I can tell you because here they just mentioned mature ribosome can pass through nuclear pore complex. Of course, correct. Only one cellular compound alone can't able to enter in the nuclear pore complex. That is your mitochondria. Okay. So why I am telling option D is the correct. That is sorry. Option D is incorrect. Why I am telling means so here you can able to find. So the correct answer for this particular question is that large molecule can be transported only if they have a cargo protein. Option D is incorrect only. I have revised many articles. So here you are able to find the nuclear export of large molecule through the nuclear pore complex such as new ribosomal subunit. So ribosomal subunit alone can able to pass through this nuclear pore complex and maturation, that is immature ribosomes alone can able to enter. Maturation of ribosomes will be taking place in the cytosol. So here they have given mature ribosome can pass through NPC, which is absolutely a wrong statement. So the correct answer for this nuclear pore complex is that large molecule can be transported. Okay. So the next question. So prolamellar bodies holding pigment precursor. So correct answer is etioplast. So etioplast or a plastid that will be formed when the leaves or other organs grow in a period of darkness. Okay, so these etioplasts will be forming as a first plastid organ when the plant or when a seed is getting germinated. This particular etioplast will be forming, and they have a unique mechanism. Like they have a paracrystalline tubular lattice like that, and they have a peripheral display membranous respectively called as prolamellar bodies and prothylakoid. So they have a pro-pigment containing material. So once the plant get grow well means this ethioplast will be converted into chromoplast, which will be holding a chlorophyll pigment. This ethioplast they don't have chlorophyll pigment, but they have few precursor molecule for providing few pigment for the plant. Next is annealing temperature of primer does not depend upon, it does not depend upon secondary structure of primer, whereas annealing temperature of primer depend upon the GC content of primer, length of primer. At the same time PCR buffer which we are employing also play a vital role in the determining the annealing temperature. Next is newborn has taken a drug that affect the thymus. Now what will be happen? So first option they mentioned there will be inability to rearrange the antigenic determinants. Second option is that T cell will not differentiate. Third option is that there will be a loss of class 2 MHC. Fourth option is that there will be a reduction in T independent antigen on B cell. So we all know the correct option is option number B, that is when the newborn doesn't have a thymus, or when he take any drug and it has affected, it makes a thymus non-functional, means and ultimately his T cells will not get the process of differentiation, because we all know T cells will be originated from bone marrow, and it will be differentiation will be occurring in thymus. Next is, which of the following does not require a prior target sequence? The correct answer is AFLP, that is amplified fragment length polymorphism. So here you can able to find it does not require any prior information regarding the target genome. Okay, so the correct answer is AFLP. Next is a unique question. It is most interesting question. I know how many of you had given a correct answer. Which enzyme is used in order to reduce or minimize the error during PCR? So correct answer is PFU polymerase. So it is also a thermostable polymerase which is employed in PCR and is isolated from this particular Phytococcus aureus. And this particular enzyme will be replicate DNA even at a temperature of 75 degrees Celsius, and it has two activities are there. It holds a 5' to 3' activities there, so that it can able to polymerize the nucleotide at the time of PCR. At the same time, it also hold a 3' to 5' exonuclease activity, that is proofreading activity. Okay, so base misincorporation that may occur during polymerization are rapidly removed by this particular proofreading activity. Whereas if you take the stack polymerase, it only holds this particular 5' to 3' DNA polymerase activity. The stack polymerase doesn't have this 3' to 5' exonuclease activity. So the error rate will be very high when you employ this stack polymerase, whereas that's the reason why many PCR techniques are nowadays they are targeting for this PFU polymerase, which also hold both the DNA polymerization activity, at the same time they also have this proofreading activity. Next, which of the following is not true regarding this DNA eukaryotic transposition? The first thing, DNA transposition has short inverted repeat, correct. DNA transposition have cut and paste mechanism. This is also a correct option. Next is retrovirus like retrotransposon have wholly 8 tail. This is also correct option. But the last option is that non-retroviral transposon, they don't have this long terminal repeat. But here they just mention it hold a long terminal repeat. So the correct answer for this particular question is D, which is not at all true. Next, which of the following is the most common error that you can observe the translation? For this question, based on my experience, I'm telling you the correct answer might be reading the codon twice, because there are no chances for to incorporate a wrong amino acid, because tRNA have this particular amino acylation process. At the same time, oval base pairing is also been there. So based on that particular two criteria, I just omitted this particular option. So reading of codon twice might be one of the most common error during this particular translation process. Next, in mutant Arabidosis, only sepal formation alone occurs in all the four olds. So what is the mutation? Okay, so both the B gene and C gene will be mutated. That's the reason why sepal formation occur in all the four odds. Okay, so there is a double mutant of for B and C. Next is co-stimulated signal for T helper cells. So the correct answer is in T cell CD4 will be present, and in APC MHC class 2 will be present. This is one such kind of co-stimulatory molecule required for activating your T helper cell. Next, your beta decay releases. So beta decay will be releasing the isobar. Next, with regarding to photosystem 1 and photosystem 2. If you know the difference between photosystem 1 and photosystem 2, you can able to easily give a correct answer for this particular question. First option they had given is that electron flow from photosystem 1 to photosystem 2. This is absolutely wrong. We all know electron will be flowing from photosystem 2 to photosystem 1. Next is these photosystems are named after the reaction the catalyst. No, it is not at all. Nowhere we can't able to find this particular statement. Next, photosystem 1 is for reduction. Of course, photosystem 1 is a strong reductant and it is a weak oxidant. Okay, so photosystem 2 is a strong oxidant. Okay, so photosystem 1 is for reducing and it will be producing NADPH. And last option is absolutely wrong. So the correct answer for this question is option number C, because last option I just mentioned both the photosystem are present in the stacked region, but we all know the location of photosystem 1 and photosystem 2 will be varying in a minor differences. Next is factor that does not increase the gene complexity in higher organism. So based on my knowledge, I'll be telling number of repeat sequences. Okay, that's a factor which will not be increasing the genome complexity in case of higher organism. See, if you want to change any answer for any particular question, you can do. I will not mind you. I will be welcoming those children who are doing correction for those kind of lectures that I am giving you. Okay, so you can add up any other statement, or if you are telling few other options are correct means, please do tell me in the comment section. It will be very much helpful. Next, they was asking about the lysosomal signal. So this time two single question has been came. Okay, so first lysosomal hydrolysis, they will be carrying hydrolysis. They will be carrying a unique marker called called mannose-6 phosphate. So this particular group will be helping for the lysosomal hydrolysis and other protein that are destined for the lysosome to get transported from the trans-Golgi network to lysosome. The correct answer is mannose-6 phosphate. I hope all are will be giving this particular correct answer for this particular question. Next, what is the difference between channel protein and carrier protein? So first thing is that channel protein are involved in both active transport and passive transport. This is completely wrong because carrier protein alone are involved in both the active transport and passive transport. Next, channel protein change their conformation. Absolutely wrong because they form a channel. There is no need to change the conformation, whereas carrier protein will be changing the conformation in open state and closed state. Okay. Next thing, carrier protein passively transport molecule. No, as I mentioned you earlier, carrier protein are involved in both the active transport and passive transport, whereas the last option is the correct answer for this particular question, which means carrier protein will be made in free ribosome, whereas the channel protein will be made in rough endoplasmic reticulum. The next question is for rate zonal centrifugation, which of the following is not used? So cesium chloride will be used, sucrose will be used, and ficol will be used. And I could able to find few important research articles saying this particular guanidinium HCl is also being employed as a gradient in this particular rate zonal centrifugation. But here the correct answer for this question is this particular answer, because some cases they will be acting as a denaturant. Okay, the correct answer for this question is guanidinium HCl, which is not used in the rate zonal centrifugation. Next is which carries a 2-carbon precursor moieties from mitochondria to cytosol during the fatty acid biosynthesis. See, fatty acid biosynthesis will be occurring in cytosol. So the acetyl-CoA need to be transferred from mitochondria to cytosol. Only then they can able to participate in fatty acid biosynthesis. So the molecule which will be carrying the acetyl-CoA is that citrate. So acetyl-CoA in the form of citrate, it will be easily transported from mitochondria to cytosol. Okay, whereas from cytosol to mitochondria, the fatty acid will be carried by another molecule called as carnitine. Okay, so yeah, in the next video, that is part 3, we'll be discussing few other important questions that has been asked in Barc examination. Thank you friends, thanks for watching this video.